Dihybrid cross and free distribution law of Mendel. Dihybrid cross is hybridization which include a couple of contrast traits. Until now, we study monohybrid cross, which crossing of two organisms for just only one trait, like red crossed with white, or tall crossed with short. But now, in the hybrid cross, we cross two organisms for not one, but a couple of contrast traits. Like what? Like round and yellow seeds with crossed with another green and ringlet, or red colored and axillary positioned flower with another white and terminal positioned flower. So in the hybrid cross, we use couple of traits for each two organisms. This is because the yellow, for example, if the pisum sativum of pure round yellow seeds are pure round yellow seeds crossed with another ringlet and green colored organism, so the all result organisms are round and yellow. Why? We know the trait which appear in F1 generation is dominant. So means the yellow and roundness is dominant over green and ringlet. If we left this hybrid organism which for two traits for cell pollination, the hybrids, they will produce number of four type of gametes. How? Like G capital, G small, W capital, W small. So we take G capital with W capital, one gamete, G capital with small w, another gamete, G small will capital W, another gamete, and G small with small w, another gamete. So hybrid organisms always produce number of gametes. But we remember when there's only one trait, hybrid produce two trait, both of them hybrid produce four kind of gametes. So in Punnett box, for male and female gametes are vertically and horizontally arranged, and we draw the Punnett box. We see there are about 9 yellow colored organisms and the others are green. So we simply look the phenotype like round and yellow, round and yellow, round yellow, round and yellow, round and yellow, round and yellow, round yellow, round yellow, round yellow. About 1, 2, 3, 4, 5, 6, 7, 8, and 9 of organisms are round and yellow for F2 generation. Ringlet and yellow, 1 and 2 and 3. Round and green, 1 and 2 and 3 organisms, and ringlet green organisms, only 1. So the phenotypic ratio, as we studied for monohybrid cross, was 3:1, like 3 tall, 1 short. But for second generation, for the hybrid cross in Mendel genetics, also the phenotypic and genotypic ratio are fixed. So phenotypic ratio like 9, 3, 3, and 1. So when we solve the question, we not reach this result like 9, 3, 3, 1 ratio means the solution is not true. Selective crossing for two traits means when we change the genotype of parents, like pure for color but hybrid for shape, so the number of organisms change with another pure recessive produce one gamete, One hybrid trait produces 2 gametes, so number of organisms also 2. So another organism which hybrid for 2 traits, 2 times 2 produce 4 type of gametes. But pure recessive again produce only 1, so the result also 4 type of organisms. So as we see how we change the genotype or number of hybrid traits, so number of organism changes. So selective crossing for 2 traits. We select what? We select the genotype of parents to find different ratio or different results for F1 generation. Interpretation of the results of Mendel in the light of the knowledge of the function of the chromosome and genes. There is no new information in these 3 short paragraphs, but like a revision about what we studied before. Studied by definition of chromosomes, this is the installation, like coiling, of composite linear shape of DNA molecule, which we call it chromatin network before. Ribonucleic acid and protein, we remember the nucleolus, which contains the coded genetic information sequence and can be viewed through the process of cell division, because chromosome can be seen only during cell division. The gene is a part of DNA chromosome that control at least a specific genetical trait. Since the chromosome present on the image matched in pairs, like rule of Mendel, we call it unit factors in pairs, it is also the genes found in image of pairs, A capital A small for a certain trait, and B capital B small for another trait, and whether the individual is hybrid but is not symmetric of these 2 traits. This, of course, supports the first hypothesis of Mendel, which we said the unit factors in pairs, but during gamete formation they separate one from another, free distribution law, and they again make pairs by fertilization. The phenotype relationship between the behavior of chromosomes and gene also through the meiosis, and it receives each gamete chromosome and each pair of identical homologous chromosomes, because during gamete formation the homologous chromosomes, which are carrier of genes or allele genes, are separate one from another. So when those gametes unite during the process of fertilization, the children receive one gene for a particular trait from father and another from their mother to form again a diploid organism. The free distribution of the chromosomes over the gametes during the meiosis supports the law of Mendel, which is free distribution law. This is because the genes being unattached. So each gene not depend on one another according to Mendel. So why? Because they are not on the same chromosome, but each gene located on a different chromosome, so they are independent and distributed through the gametes independently. Application on Mendel's second law. A Drosophila melanogester of ebony color with long wing pollinated with a gray and vestigial. The first generation were gray and long, knowing that the genetic factors for the traits are located on 2 different chromosomes, and ask about the A, B, C, D questions. In genetical problems, what we need to do at first to read all the question and focus on the given. So what is the genotype for the parents and members of first and second generation? Phenotypic ratio for second F2 members, and their ratio, genetic ratio for F2. Show the type and number of units which possible between the gametes. Clarify that on the Punnett box. As we look like a whole, it asks about the parents, F1 generation, kind of gamete, F2 generation, their phenotypic and genotypic ratio. So this question contains all the questions or probabilities can be asked in a questions. When we get it fully, we can solve any question about Mendelian genetics. But a Drosophila melanogester for ebony color with long wing, pollinated with another with a gray and vestigial. So we understand it's Dihybrid Cross. So what we need to do first, we predict the genotype of parents. Right, by ebony color, long wing with another pollinated gray and vestigial. Parents, we know their phenotype, but we don't sure about their purity, and we don't know information about which trait is dominant, which trait is recessive. So we not have to memorize any trait about their dominant and recessive in Mendelian genetics. But at the first generation, gray and long. So all the products of F1 generation, gray and long. When I said all the product, means parents are pure. Now we know both parents, for both traits, they are pure. In second step, we predict which traits are dominant, which one recessive. So the traits which appear in F1 generation are dominant. Like which traits we have? We have ebony, we have gray, we have long wing, and we have vestigial. So Miss gray is dominant over ebony, long is dominant over vestigial. But in book it uses the opposite letter, so we use the same as book. So when we write the given, we can choose any letters. So we use capital E for gray, small e for ebony, capital L for long, small l for vestigial. So ebony color, small e, long wing, capital, with another gray, which is dominant, E capital, with vestigial winged, short, small, and small l. So how did we get this result? How we know which trait is dominant, which one recessive, and how we know the parents are pure? We think and we then find, we use the given information. So in ministry exam, we have to write this information and we call it conclusion. How? So the first generation, all gray and long. So all when there are only one kind of organism in F1 generation, mean the parents are pure for all traits. One. In second, because F1 generation, all of them are gray color, means gray is dominant over ebony. We show gray with capital E, ebony with small. And because all the products are long winged, long is dominant over vestigial. We show long with capital L and vestigial with small. It's our conclusion. So let's find the F1 generation. Parents, pure for both traits, produce one kind of gamete, one from E, one L. Also pure, capital E, small l, pure F1 generation formed by union between male-female reproductive cell, capital E, capital L, small l. So F1 generation, genotype 100% hybrid, gray color, long-winged, Drosophila melanogester. For F2 generation, in F2 generation, there is no information about the parents, so we get the members of F1 generation. We left it for what? Self-pollination. So P2, E capital, E small, L capital, L small with another E capital, E small, L capital, L small. Gametes for second generation, E capital with L capital, E capital with small l, small e, L capital, small a, small. That's 1 and 2 and 3 and 4. For second case also similar genotype produce similar kind of gametes 1, 2, 3 and 4. As we see, there are 4 gametes, 4 male and 4 gametes, 4 female. So we draw what? We draw punnet box. 1, 2, 3 and 4. 1, 2, 3 and 4. The horizontal line for female, vertical line. For male, we write their genotypes, capital E, capital L, capital E, small a, small e, capital A, small e, small l, capital E, capital L, capital A, small e, capital L, and small a, small l. F2 generation, by crossing between male and female gametes, we draw our punnet box, and now we can see F2 generation. It asks about, we have the genotype and phenotypic ratio for F2 generations. So for a gray color and long winged, 1, 2, 3, 4, 5, 6, 7, 8 and 9, means 9 organisms are gray color and long winged. For gray color and vestigial, 1 and 2 and 3 organisms. For ebony color and long winged, 1 and 2 and 3 organisms, and 1 organism, ebony color, vestigial, winged. So 9, 3, 3, 1, it's phenotypic ratio for Mendel, which is fixed. Then, if we look at the genotypic ratio, we look full capital E capital, E capital, L capital, L capital. How many organisms like 1? For in second capital, 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 small, how many? 1 and 2. So capital, small, capital, capital again, 2. Capital, small, 4. And like this, we look the exact about the genotype of organisms, and we find ratio like 1, 2, 2, 4, 1, 2, 1, 1. Like a ratio which are fixed in Mendelian genetics. Related to topic question 7 from chapter questions, the Genian pig with rough and black hair and black hair were crossed with a female with rough and white hair. They had a number of births as the following: 3/8 rough and black, rough and white, smooth and black, smooth and white. What are the genotypes for the parents and the product individuals? The traits of the rough hair and the black color is dominant, means rough dominant over smooth, small r. Black is dominant over white, small. So rough and black dominant, rough and white dominant and recessive. But because the F1 generation has different ratios, means the parents are hybrid. So to be sure about the purity of parents, we look the individuals. When we look individual with one rough organism crossed with another rough, there are rough and also smooth. Remember the test cross, if all the products are same, means they are what? Pure. But when there are different probabilities, means they are hybrid. So the smooth organism, like smooth black, means what? Smooth means small r, small r. So two organisms which is rough and rough cross it, but they find the smooth. The genotype of smooth is small r, small r. We know this individual receive one of each allele from one of the parents. So two dominant trait crossing produce one recessive pure, means parents, both of them are hybrid. So hybrid and hybrid. For second trait about black and white. White, we sure it's pure. Why? Because recessive always pure to be seen in phenotype. What about the black? So black and white crossed, if they are pure, must be one kind of organism, all of them black. But even if they are white, means black also hybrid. So we see white organisms too. So white also means B small, B small. We know exactly one of them received it from mother, but what about the second? It received it from the parents. So what we think, what we did now, it's only conclusion. So a question asked about what are the genotype for the parents. Now we found the genotype of parents and the product individuals. Parents produce gametes R capital, B capital, R capital, B small, R small, B capital, R small, B small, 1 and 2 and 3 and 4. For female, R capital, B small, R small, B small. Two kind of gametes. So we draw what? We draw our Punnett box. For what? Female gametes and male gametes. R capital, B capital, R capital, B small, R small, B capital, R small, B small. For female gamete, R capital, B small, R small, and B. So at the end of crossing between male and female to find the F generation, we reach the result of 1/8 smooth and white, 1/8 smooth and black, 3/8 rough and white, rough and white, rough and white, and 3/8 rough and black, rough and black, and rough and black.